session we have seen about the nuclear membrane organization, and now we are going to see about the cell cycle. Have you ever watched the growth of butterfly? If so, probably you might have some idea on cell cycle. Butterfly go through some transitions from worm into pupa and finally into a butterfly, right? Similarly, for the growth and development of an organism, the cells must undergo several series like birth and reproduction. Let us move into the module 4, cell cycle. Cell cycle is the process where the cell grows and development into new cells, and also it divides the DNA. Cell division is the process where a single cell divides into two identical daughter cells. In humans, this process takes up to 24 hours, whereas in yeast it takes up to only 90 minutes. Based on the organism, the time duration for cell cycle and cell division varies. They perform these tasks in an organized series of cycles where a daughter cell go through these same process from first initial stage. In an eukaryotic organisms, it goes two phases. One is interphase and another one is mitotic phase. In interphase, the cell grows and make a copy of its DNA, whereas in mitotic phase, the cell divides its DNA and also its cytoplasm. Finally, it gives birth to two identical daughter cells. Coming to interphase. Interphase is the longest stage in the cell cycle. In this, the cell grows by acquiring nutrients, molecules and proteins. It starts the cell cycle process by replicating the DNA. It occurs in three phases. One is G1 phase, which is also known as first gap phase. Next is S phase, which is also known as synthesis phase, and finally G2 phase, which is known as the second gap phase. In the G1 phase, the cells grows and do its normal function and replicates the necessary organelles. Next, it moves to the synthesis phase where the DNA replicates. Finally, it moves to the G2 phase where the cells grows and prepares itself for the cell division process. After the G1 phase, sometimes it moves to the G0 phase, which is the resting phase. We all know cell cycle is a complicated process, right? In order to make sure everything goes right, we need some checkpoints at the end of each phase. At the end of G1, they check for the size, nutrients, molecular signals and DNA integrity. At the end of S phase, they check for the chromosomal spindle attachments. And finally, at the end of G2 phase, they check for the DNA replication and DNA integrity. If all they are cleared, they move to the next phase. If any malfunction occurs, the phase stops over there and starts from the initial stage. If malfunction is not repaired, they may lead to the genetic disorders, like also it makes to the cancer cells. Before entering into the mitosis process, we will discuss about some real-time examples. You are cutting your hair, it grows again, right? You are cutting your nails, also it grows again. If you face any injuries in your hand, it repaired within few minutes or few days, right? It all because of this process. Mitosis is the process where a single cell divides into two identical daughter cells. It occurs in several phases like prophase, metaphase, anaphase and telophase. It usually occurs in the body cell. We are going to see about the mitotic phase one by one. In prophase, the chromatins condenses to form chromosomes. The centrioles moves to the opposite poles to form mitotic spindle fibers. In this stage, the nucleolus and endoplasmic reticulum starts to disappear. In metaphase, the chromosomes lined up at the center in attachment with the mitotic spindle fibers to form the metaphase plate. Here in this stage, we can view the chromosomes very clearly. Also, in this stage, the nuclear membrane completely disappears. In anaphase, the chromosomes which are attached with the mitotic spindle fibers moves apart to the opposite poles. In simple words, chromosomes have four arms, two short arms and two long arms. One short arm and one long arm forms the one sister chromatids, right? So here one sister chromatid moves to one pole and another sister chromatid moves to the opposite pole. Now we are going to see about the telophase, which is the last phase of mitosis. In this phase, the nuclear membrane and cell organelles starts to reappear. Here the cleavage furrow forms at the center which initiates the next process called cytokinesis. Cytokinesis is also known as cell division, where cyto means cell and kinesis means divisions. In this cytokinesis process, the cells has to divide continuously to produce new cells. For this stage, initially the division of chromosome occurs. Later on, cytoplasm and cleavage furrow divides to form two new cells. Mitosis play a very important role in our body. It plays a vital role in the growth of organs, and also it repairs the cells, and also it repairs the wounds. It plays a very important role in the apical meristem for the continuous growth of plants. Majorly, it gives birth to the new daughter cells with similar genetic information. We are going to see about the another type of cell division, that is meiosis. What we have seen in the mitosis process, a single cell gives birth to two identical daughter cells, right? And in meiosis, a single cell gives birth to 4 haploid cells containing half the amount of original genetic information. I think you are familiar with these kind of talks, like you look like your father and your character resembles your grandfather or grandmother, etc. These talks are only because of this meiosis process. Meiosis is divided into two types. One is meiosis 1 and another one is meiosis 2. In meiosis 1, pairing of homologous chromosomes occurs and recombination also occurs. Okay, tell me, what is homologous chromosomes? It is nothing but it consists of one maternal chromosome and one paternal chromosome with similar appearances. And in meiosis 2, 2 diploid cells gives birth to 4 haploid cells. Now, we are going to see about the meiosis 1. It consists of several phases like interphase, prophase 1, metaphase 1, anaphase 1, and finally telophase 1. Interphase we have already seen in the mitosis phase, and now coming to prophase 1, it has several series: leptotin, zygotin, phagotin, diplotin, and finally diakinesis. In leptotin, the chromosome condenses, and in zygotin, pairing of chromosome occurs to form synapses. In phagotin, the paired chromosome exchanges to form crossing over and recombination, and in diplotin, the paired chromosome separates. Finally, in diakinesis, the nuclear membrane degenerates and the spindle fiber develops in preparation for homologous chromosomes. Now, it is metaphase 1, where the homologous chromosomes lined up at the center in attachment with spindle fibers to form metaphase plate. And in anaphase 1, the homologous chromosomes moves apart to the opposite poles. And finally, in the telophase 1, the nuclear membrane and cell organelles regenerates, which leads to the cytokinesis process. At the end of cytokinesis, we get 2 diploid cells from a single cell. Coming to meiosis 2, it consists of several phases like prophase 2, anaphase 2, metaphase 2, and telophase 2. It is completely similar to the mitosis process. At the end of the meiosis 2, we get 4 haploid cells from 2 diploid cells. Like mitosis, meiosis also playing a very important role in our body. It plays a very important role in sexual reproduction. Due to crossing over and recombination, it plays a very important role in new traits and new generations. Okay, we have seen about the mitosis and meiosis process. Now, let us discuss about the differences between them. Mitosis have 4 stages, whereas meiosis has 8 stages. Mitosis undergoes body growth and organ growth, etc., whereas meiosis undergoes sexual reproduction. Mitosis plays a very important role in wound healing, etc. Meiosis don't play that kind of roles, but meiosis plays a very important role in genetic variations due to crossing over and recombination. Now, we come to the conclusion of this module 4. Here, we understand about the cell cycle, cell division, meiosis and mitosis process. In the next module, we are going to see about the basic chemical constituents of living cells. Thank you.